Welcome, everybody. Good evening. It's a great pleasure to have everybody here to kick off the 2025-26 FBRI BTC Maury Strauss Distinguished Public Lecture Series. I want to start, as always, of course, to remind everybody how incredibly grateful we are for the legacy of Mr. Maury Strauss, who supported this program to bring the world's innovators and thought leaders in science and medicine to our community. And to Maury's family who are here, we thank you very much as well for your ongoing support for this program. It means a lot to us. So tonight's introduction is a two-pager, I'm sorry, instead of a one-pager, and I'll explain why, but I'll try not to delay too long. So this evening's lecture coincides with the 15th anniversary of the Fralin Biomedical Research Institute at VTC. It originally opened in 2010. And on cue, Heywood walked in. This is great. I was just going to say it was originally called the Virginia Tech Carillion Research Institute, but after a transformational gift from Heywood and Cynthia Fralin and the Fralin Family Endowment, we changed the name to the Fralin Biomedical Research Institute at VTC. And it's been an exciting run so far, to say the least, with many of the originally conceived goals and milestones having been achieved and surpassed, I would say, due largely to the ingenuity, dedication, and commitment of the exceptional teams of faculty, students, and staff who work here. In addition to the Research Institute's impact on the local economy that's approaching $2 billion recently, the scientific advances made by the teams of researchers here are accelerating our understanding of and our ability to change the course of diseases, including cancer, heart disease, and neuropsychiatric disorders, as well as to prevent disease and to help people live healthier lives. In many cases, these innovations are moving from the labs within our university environment at Virginia Tech into applications being made available to our clinical partners, including Carillion Clinic, as well as the greater community, delivering on the promise that was made more than 15 years ago. It is no secret, however, that we are living in a time of uncertainty with regard to our nation's ability and will to remain preeminent globally in the biomedical and other research enterprises. However, I am confident that we will continue to lead the world and change lives for the better, including through the work occurring right here in Roanoke. It is now my great pleasure to introduce someone who gives me the confidence to make that statement, and who's been at the very heart of the program from nearly the very start and continues contributing mightily to our mission in so many important ways. A quintessential scholar, scientist, medical innovator, entrepreneur, and gentleman, Dr. Rob Gourdie. Rob is the Heywood Fralin Professor here at the Fralin Biomedical Research Institute, where he also leads our Center for Vascular and Heart Research. He is also a professor of biomedical engineering and mechanics in his brand new chair. Miguel is here somewhere, there he is, from Blacksburg. And he's the Commonwealth Commercialization Eminent Scholar. Rob joined the FBRI in 2012, coming to us from Charleston, South Carolina, where he was at the Medical University of South Carolina, and he was professor and Board of Visitors Eminent Scholar of Regenerative Medicine there. Previously, he was a British Heart Fellow at the University College London. Rob grew up in New Zealand, where he studied cell and molecular biology at the University of Auckland, and he completed his Ph.D. in biophysics at the University of Canterbury. His many original and insightful contributions have earned him global recognition, including being elected as a senior member of the National Academy of Inventors, being elected as a fellow of the American Institute of Medical and Biological Engineers, receiving Virginia Tech's first ever R35 Outstanding Investigator Award from the National Institutes of Health, and receiving the Tibbetts Small Business Administration Prize presented at the White House, and multiple entrepreneurship awards. Rob's careful, thorough, and bold approach to unraveling some of the most fundamental processes in living cells in his quiet but powerful way is changing our world for the better. His journey of curiosity, planning, designing, and implementing definitive experiments, never being satisfied until he has advanced the work into the real world of human health, is a shining example of what can be when we unbridle the beauty of scientific inquiry. Rob's very first series of studies revealed in detail the localization and distribution of previously poorly understood communication channels called gap junctions between heart cells and how they form during early development. This work has had profound implications for understanding cardiac function and disease. Fast forward 35 years with over 200 publications, with 66 students and trainees working under his able mentorship, and Rob and his team are developing new therapeutic approaches based on early molecular analysis of those channels that he had done, and including entirely new process to treat a range of disorders, including but not limited to heart damage. Moreover, he has boldly taken his insights and applications where few academics dare to tread, into the marketplace for the benefit of us all. Please join me in welcoming Dr. Rob Gourdie as we hear about this incredible thing.
Hi everyone, welcome, and also welcome to all those that are online. Mike, thanks for those very kind words. I'm going to say some words about you in a second as well, so you're not getting away with it. All right, let's get cracking. Okay, so I did want to talk about leadership just right out of the gate. I have been very fortunate over my entire scientific career to have some very able leaders head the scientific enterprise that I've been part of. Now, anybody who's inside science knows that scientists present an HR problem. They're often unique individuals, and so wrangling those individuals takes a special skill set. This is my chairman in Charleston, Roger Markwold. Wonderful leader, very supportive. Of course, here's my boss here, Mike Friedlander. And I've noticed a few things about scientific leaders, effective ones. Of course, they're energetic, they're wise, they're generous, but also they actually like people, including other scientists. And that's probably one of the least valued or least recognized skills of a very effective scientific leader, is their ability with people and actually enjoying other people and what they do. So yeah, I've been lucky to have very able leadership. I also, right up front, want to recognize my lab. I'm going to tell some stories about the work that we do, but I think, I'm just privileged to work with some remarkable people, particularly remarkable youngsters who are at the beginning of their careers, bright, creative, driven. We put a lot of emphasis on our football team at Virginia Tech, as not so good as it is, but these youngsters are stars in their own right, even if they're not quite as well recognized. All right, so let's start with some basic facts about disease. So this is a chart of causes of death over the last 100 years or so. And you can see right at the outset, here's heart disease. It's one of the biggest causes of mortality in humans for a very long time. Obviously, cancer is a big problem too, and here's stroke. So these big three, really account for a lot of the mortality that we encounter. So heart disease and heart disease and cancer, over 500,000 individuals per year will pass away from them. Stroke's not quite as many people, but still a significant number. So together, around 1.5 million lives per year are lost to each of these three causes. So in this talk, I'm going to try and share some of my journey in basic cell biology. And I think, we are working our way towards new approaches to understanding the causes of these diseases and perhaps new ways of treating them as well. All right, so Mike mentioned gap junctions. So what are gap junctions? Well, gap junctions are literally connections between cells. They're tiny channels that link the inside of one cell to its neighbor so that it can pass small molecules from the inside of that cell to its neighbor. And so if you're thinking of a metaphor for what gap junctions are, they basically serve as the internet, or at least the hardware for the internet that links tissues, organs, etc., together. They connect cells, enabling rapid, direct sharing of information. Now, here's another depiction of the structure, and you'll see that the gap junction is comprised of an aggregate of these channels. You'll also see how one channel from one cell is opposed to a channel from the neighboring cell, and within this two-channel structure, you have a pore, a channel which links the two cells. Now, of course, in my own particular style, I haven't focused much on the gap junction itself. Our work is largely focused on the edge of the gap junction, in a region that we call the perinexus. And within this region, the individual channels, which will eventually dock with channels from the neighboring cells, are kind of held in place by a scaffold that's linked to the cell cytoskeleton. And we showed in a series of publications how this is important in regulating the size, the amount, the position of the gap junctions between the cells. All very important, but very fundamental cell biology. Now, the perinexus itself was co-named. This is my graduate student from a number of years ago, Matt Rett, by myself and he in this paper in 2011 on molecular biology of the cell. And basically, the discovery and the characterization of this structure sealed my fate. And we've gone on to work on the role of this structure in cardiac bioelectricity, largely in collaboration with my friend and colleague, Steve Pulsing. Steve was down here a second ago, but now he's disappeared. There he is. But also the role of perinexal constituents, connexin 43 and ZO1, in injuries to the heart and other organs. And I'll get into that as I get deeper into the talk. So let's talk about the perinexus and cardiac bioelectricity. So of course, this is my little joke, the heart's an electric organ. It's not really, it's kind of a drum really, because it beats out a rhythm, right? But electricity or electrical activation in the heart gets converted to the mechanical pump function of the heart, which of course displaces the blood and moves it through the body. But this electrical activation can go wrong. So in this GIF here, for example, we have a depiction of a very common arrhythmia that occurs in humans, atrial fibrillation, where, and I'm sure there's a large number of people that are sitting in this audience that have to put up with atrial fibrillation. It's an uncomfortable and often difficult thing to deal with. But basically, the atrial appendages quiver, and it's a type of arrhythmia. Now, of course, the more dramatic arrhythmias, such as of the ventricle, that can lead to sudden cardiac death, get a lot of attention. But the fact is that really over the last 2 or 3 decades, we've had very few new treatments for arrhythmias. There's many reasons for this, including they're dangerous, and that kind of makes pharmaceutical companies wary of developing drugs to treat them. They don't want to be sued, etc. But perhaps one of the big reasons that we don't have new medicines for this condition is we really still don't understand how the heart's electrical activation works. And just to emphasize, coming back to the atrial fibrillation, it's not just a cause of discomfort. Unfortunately, atrial arrhythmias account for about 20% of stroke deaths each year. The clots that form in fibrillating atria break off, move into the brain, and often lead to devastating circumstances. So addressing this problem is atrial arrhythmias, but also ventricular arrhythmias, is a real gap that needs to be addressed. Okay, so electrical conduction, what's it all about at the cell level? So this is a little picture I laboured over this week. But the way that electricity flows between cells in the heart and another excitable tissue, in science or biology, we refer to tissues that have electrical activity as excitable, was thought to be fundamentally different. And I've kind of simplified the circuit here a little bit, but we can imagine it working a little bit like this. And this is the old-fashioned way of thinking about it. That basically you had charge in the downstream cell, and it flowed across the gap junctions to the upstream cell to initiate the activation of that cell. Which of course is quite different from how we conceive of electrical activation for the most part, moving in nerves, where the electrical activation jumps across this very tiny gap, the synapse, via chemical signals, which then reinitiate the electrical activation in the downstream cell, which then propagates away. So this synapse is a very narrow gap of extracellular space. Okay, so just keep that narrow gap of extracellular space in mind. Okay, so we can isolate individual heart muscle cells. This is a heart muscle cell, I believe, from a mouse, maybe a rat, I can't remember. And it's been stained with a dye that picks out the calcium in the cell, and we can see the activation wave. Some of these cells beat in culture, so you can watch their electrical activity. And here we can see we'll have to make it go again. There we go. The wave of activation moving along the cell. And if we capture it with the electrode, we'll see something like this. You might have heard of this electrical structure. It's called an action potential. And basically, it's got a number of different parts to it, but the part that we're interested in is this first part here, because this is where the electrical activation initiates. And what dominates the electrical activation of this phase of the activation? It's a bunch of highly conductive, highly regulated channels called sodium channels. Different type of channel from the one I was telling you about, the gap junction channel, but nonetheless a channel. Okay, so one of the questions that I started asking myself and my crew 10 to 15 years ago was, what was the relationship between the gap junction and the sodium channels? It's known, and it was known that sodium channels were expressed in the intercalated disc, but also around the outside of the cell. But I wanted to know what it looked like in relation to the gap junction. So here we have a gap junction, this aggregate of channels. We've labelled it with an antibody that's with a fluorescent tag on it so we can see where it's at. And when we label the sodium channels, we can see that actually they have a very intimate relationship with the edge of the gap junction. In this very domain, over this very domain, in fact, the perinexus, that sits at the edge of the gap junction, and where I was telling you this scaffolding molecule, the hemichannels, etc., are located. And I've picked out this narrow gap of extracellular space with a marker pen. When you look down on an electron micrograph, you don't see a red domain. This is me drawing over the top of it. But basically, these channels sit right here and right here. And of course, just to emphasize the analogy I'm trying to make, this cleft of extracellular space is about 15 to 30 nanometers wide, which is unbelievably narrow. It's sort of, you could split a hair, 6 to 10 times to get down to that kind of thinness. And it's about the same width as these synaptic clefts that you find between nerve cells. Now, I'm trying to make an analogy. I'm not saying that they're the same, but, the similarities are striking. So we're on this path of looking at the edge of the gap junction and asking questions, what else is there? And this young man here, Sai Veeragarvan, he joined me as a postdoc in 2012. He actually trained with Steve Pulsing. And I also have to say that Steve and I worked closely together as we progressed along this path. This is a technique called super resolution microscopy. I won't get into too much gap junction. And here is this halo of sodium channels around this particular gap junction. So again, without going into too much detail, what we built up was a model that looks something like this. So here is the gap junction with our clusters of channels, and then right next door to it, in the perinexus, we have these sodium channels. But there's something interesting about these sodium channels. They almost have a junction-like structure themselves. Here you can see the pore of the sodium channel, but it's linked across the extracellular space to a partnering sodium channel in the other cell via this adhesion molecule. which is actually part of the sodium channel complex itself. It's called the beta subunit of the voltage-gated sodium channel. And what we noticed and what we fixed on, and others noticed it as well, is that the adhesion subunit has what's called an immunoglobulin domain on it, which is famous for mediating adhesive interactions. And, to a scientist or someone looking at something like this, your first reaction is, what is a channel doing with a adhesion molecule associated with it? But, after we'd sort of looked carefully and figured it out, this was the arrangement we deduced. So one of the things my lab does is develop peptides to target the activity of proteins. And what we did was we synthesized a peptide that corresponded to the sticky bit of the immunoglobulin domain, a peptide that we call BAD-P1. Now, I didn't name it, Cy did. I didn't think it was a good name, but he thought it was funny, so we went with it. And what we found when we perfused it into beating guinea pig hearts was, sure enough, here's the perinexus. Again, I picked it out with a yellow marker pen so you can see where the extracellular space is. But when this peptide went into these animals' hearts, you can see the perinexus basically gets blown apart. It destabilizes the structure. We measured it and it was significant. We also shared the peptide with a colleague at Imperial College London who uses a special technique where she can poke a patch pipette, an electrode, into the junctional space between myocytes, and we measured the loss of these sodium channels from the junctional space over time. And you can see the channels very quickly flow away from the perinexal area following treatment with this peptide. And then we also looked at the propensity for these hearts to develop arrhythmias. And you can see that as the concentration of the peptide increases, the amount of this arrhythmia called ventricular tachycardia shows a corresponding increase. So we had a peptide that disrupted the adhesion of the perinexus. We had a peptide that also caused the sodium channels that are normally locked in place at the edge of the gap junction to float away. And when this happened, we saw the heart become arrhythmic. And so, I just have to really give credit to Steve here. Steve has really gone on to define the field and in particular the role of this de-adhesion at the edge of the gap junction, the perinexus. And I'll just point to one particularly important result. He's shown that loss of adhesion in the perinexus is associated with atrial fibrillation, the most common conduction disturbance in humans. And both he and I have got a lot of pushback for our ideas because we've disrupted sort of 50 to 60 years of locked-in thinking. Everybody wants the gap junction to be responsible. And here's us, a couple of nobodies in the Appalachians saying, no, you got it wrong. But anyway, I think slowly but surely we're starting to win the crowd over. But yeah, Steve's also pushed this into understanding how pacemaking occurs. That's another story, and he'll tell it one day, probably this forum. Okay, but the point is that this perinexal structure has an important role in mediating ephaptic. And this is a word I haven't used yet. But ephaptic is basically the indirect mechanism of action potential transfer, almost like an electrical synapse that involves this tiny gap of extracellular space, as opposed to the previously understood direct transfer via gap junctions, which we don't discount as not important, but nonetheless, we think that the role of this ephaptic mechanism has been underestimated. And one of the things we've tried to do is to get this down to its most elemental nature. And this young man here, when I talk about remarkable colleagues, Zalbo is indeed a remarkable colleague. He was born in China, and he came over here to do his Ph.D., again with Steve. He joined me as a postdoc. And basically, Zalbo has figured out a way to isolate single myocytes and pairs of myocytes with intact intercalated discs, which means they have intact gap junctions and perinexi, and figure out this ephaptic mechanism at its most fundamental nature. And just to zoom through his contributions, he shows that the proteins we're interested are present. the different components of the sodium channel, as well as the gap junction protein connexin 43. He's shown that the perinexus occurs within the intercalated disc of these cells, and it responds to our peptide by becoming wider. But this is his most important insight so far. It's a little bit complex to describe what's going on here, but basically when we poke a patch pipette into a single myocyte and isolate the activity of the sodium channel, we do so by testing the activity at different what are called membrane depolarizations. We get a set of curves that looks something like this. And if you just focus on this red curve, we've isolated it here. And this is basically what we call the pre-peak. It's just before the sodium channel reaches its peak level of activity. I think it's around minus 80 millivolts. But anyway, this pre-peak curve, when we apply a mathematical treatment to it called the first derivative, and I know that sounds a bit fancy, but the way to think about it is acceleration is the first derivative of velocity. It's basically the rate of change of the curve. We see a relatively simple structure. Now, when we look at the situation in the paired cell, we find something different happening. And of course, one of the main differences is we now have these aligned junctional sodium currents that weren't present in the single cell. And when we apply the first derivative to this curve, we see something very interesting. The emergence of two troughs. Now, I know it looks like a couple of squiggles on a screen, but I can't emphasize how important I actually think this is. Because basically what Szabo has done is he has boxed this apoptic perinexal mechanism into a corner. And there it is, you know. But that's not the only part of the story I'm going to talk to you about. We call this the junctional sodium current. We're still arguing amongst each other about exactly what we're going to call this. Szabo has some ideas, I have some ideas, Steve has some ideas, but provisionally let's just call it the junctional sodium current signature. Now the other thing he discovered was when he blocks the gap junctions between this pair of cells, he basically converts the junctional sodium current signature from something like this to making it look like what you'd see in a single cell. Now, notice the sodium concentration here. It's 25 millimoles. That's about a fifth of physiological sodium that circulates in the blood. It's a non-physiological sodium concentration. We have to do that because we can't record at higher sodium concentrations because the signal becomes overwhelming from the channels. However, we can bump it up a little bit, and look what happens when we do that. Bang, the junctional sodium current re-emerges. And here is the derivative structure. So, we're just I think we're just at the beginning of, feeling our way through the significance of this, but I think what we can conclude is that the extracellular sodium is critical for activation of this junctional sodium current. And we can imagine also when physiological sodium is moved up to its 135 millimole concentration, these junctional sodium currents will be something like a lightning strike. And Steve did a back of an envelope calculation while we were sitting having a cup of tea in my office this afternoon. And he figured out that it's not just a lightning strike, it's probably anywhere between 10 to 100 times the energy density of a lightning strike within this tiny volume of extracellular space. So if you ask yourself the question, how is it that the heart beats 2 to 3 billion times in my lifetime without fail? We don't think it's because the charge is seeping through gap junctions. You have this bang, tiny discharge of energy that basically propagates the activation onto the neighboring cell. Now, I don't know if we've got any neuroscience. Steve's up there. Neuroscientists in the audience. But, I just want, and, we have a large number of neuroscientist colleagues at the Institute. But when we talk about propagating activation, we're also talking about moving electrical fields. And here's a kind of simplified, I like this GIF because it shows two magnets moving with respect to each other, but it also shows how this movement shapes the electrical fields associated with these moving magnets. Now, there was a great paper a couple of years ago from young Kuchera in Switzerland who generated some mathematical models and showed that the perinexus almost acts like a lens, focusing the field lines along which the ions flow. And so, oh, what did I just do? Yeah, okay, I'm sorry. This is what happens when I get excited. Yeah, and so, you know, I'm trying to be a little bit provocative here, but there was also a paper a couple of years ago by this guy, McIver, who suggested that the ability to focus electric field lines is key to understanding how sentience and consciousness emerges. And there's also quite a lot of published data, some of it coming out, you know, really recently, that suggests that effective mechanisms underlie brain oscillations like theta waves, epilepsy. Nice paper just in the last month or so on memory engram formation, and also that effective conduction may give a context for understanding how the binding problem works. What is the binding problem? It's the problem that people who study consciousness, when they try and understand how perceptions, thoughts, etc., are all integrated into a single field that we experience as consciousness. So, as I say, I'm trying to be a little bit provocative here. I think, you know, we can all learn something from each other, but maybe the cardiac biologist can teach the neuroscientists just a little bit. All right, so let's move on to the work on injury. So here's the connexin 43 protein, as us biologists imagine it folded in the membrane. This domain of the protein here is actually inside the cell, and the domain of the protein that we've been interested in really for the last 20 or 30 years is the very end of the protein, the carboxyl terminus, as we call it, and in particular this last 9 amino acids. We call the sequence alpha-CT, and we've made a couple of synthetic peptides based on this sequence. This was the first one we made. Here's the connexin 43 sequence, the gap junction protein sequence. We attached it to what's called a cell penetration peptide. Because the targets of this domain are inside the cell, we need to get the peptide when we apply it to cells through the membrane, and so hence the cell penetration peptide. And again, I have to emphasize that we originally made this peptide for very basic fundamental cell biological research. We wanted to uncouple the gap junction protein connexin 43 from the scaffolding molecule CO1, and then ask the question, what's happening to the gap junction? Is it getting bigger? Is it changing its position in the cells, etc.? However, I had this young man join my lab in around 2004, Gautam Gatnikar. As you can see, he's a vet, which means he didn't serve in Iraq. He went to veterinary school, and that's a silly joke. But he just finished his Ph.D. at NC State, and so he had a clinical interest. And what I started him on was looking at the migration of cells in this particular culture model here. In this model, we grow a lawn of cells, and then we put a scratch in it with a pipette, and then we watch the rate at which the cells migrate back into the scratch. It's a kind of cell culture scratch injury model. And Gautam fairly quickly discovered that the peptide caused the cells moving into the scratch to accelerate in really what looked like a dose-dependent manner. He also started working with small injuries in the skin of mice and found that the peptide encouraged accelerated the rate at which these wounds closed, reduced the inflammation associated with healing, and also reduced scar tissue associated with the healing a little bit. So he'd been with me for about 18 months. He told me he didn't want to be a postdoc anymore. He wanted to leave, start a company, and pursue the clinical development of the peptide. And so he led the peptide. He led this clinical development program through 3 phase 2 clinical trials, 2 for chronic wounds like diabetic foot ulcers and venous leg ulcers. And he also looked at the ability of the peptide to reduce scarring following surgery. In this case, he was looking at laparoscopic, very kind of small incisional wounds and looking at the level of scarring associated with it. All 3 clinical trials returned positive results. And I had a biomedical engineering student from Virginia Tech, Jade Montgomery, work with me to try and figure out what the mechanism was here, and she discovered that the peptide, one of the effects of the peptide, probably the main effect, was to change the organisation of the collagen in the scar tissue. And this was the variable that was most associated with the reduction in scarring that we noticed. And we published this in the FAHA journal a few years ago now. Now, First String, the company that Gautam and I formed together, got taken over by a private equity group and renamed itself Equal Bio. Just this year, they closed on a $140 million deal with a major pharmaceutical company. Now, when I say $140 million, I didn't see any of that money, so don't worry. But nonetheless, the company has done well. Now, one of the things that Gautam, and I really give him credit for this because he's got great intuition. It's hard to get him to work at the bench when he was a postdoc, but he always came up with the best experiments. Well, not always, but he had a gift. One of the things he intuited was that the peptide might be particularly effective in countering radiation injury. And so, together with Carissa James, who is a former Virginia Tech graduate student, she was with Jamie Smith, graduated a few years ago, and they embarked on clinical trials in animals under the FDA animal rule. And one of the initial studies they did was they irradiated pigs and then looked at the effect of the cutaneous radiation injury of the peptide over time. And in this particular experiment, they got a pretty profound reduction in the severity of the wound following treatment with the peptide. So this really caught my attention. I thought, well, skin's all very well, burns are a big problem, but, what say we could provide radiation protection for internal organs? So radiation therapy is probably one of the main tools used by cancer doctors to treat cancer. Around 50% of cancer patients receive some type of radiation therapy. But, the side effects from radiation therapy can be pretty awful. And in some cases, people don't complete their therapy due to these side effects. So there's a real unmet need to counter the effects of radiation to internal organs such as the brain, gut, and lungs. So we set ourselves this task of trying to address this unmet medical need. Now, this next slide, I have to give you a warning. But of course, the other thing is we really don't have any treatments for ionizing radiation per se, which might occur, God forbid, in the case of a nuclear attack or a nuclear accident. People talk about handing out, what are they? It's a placebo. It's not going to do much good. So there's also a military strategic need to develop radiation countermeasures. But there's a problem with this dual goal of trying to improve therapy for cancer patients, but also trying to counter the effects of high doses of radiation. And it's this: that basically short linear peptides in isolation are very unstable in body fluids. And this is a study I did with my friend and colleague down on main campus, Jia He. He works in the veterinary school. And we looked at the stability of the peptide in isolated blood, plasma, but also in living animals. And what we found was that the peptide was degraded within minutes, which is not long enough for it to have a therapeutic effect when you put it inside an animal. It works if you put it topically on the skin, but inside an animal, it's just not going to work. So I started to think about how we might deliver the peptide to a patient, whether it be a veterinary patient or a human patient, in a way that it was stable. So this is Spencer Marsh. Now, as Spencer has been absolutely central to this part of the journey, another remarkable individual. He joined me in 2020, just before the pandemic broke out. He begged me not to send him home to write grants, but instead declare that he was an emergency worker, so he could work in the lab, which he did. Perhaps I should have let that piece of information out, Mike, I don't know, but we're through it now. But anyway, we just a little bit about exosomes. And the rest of the story really, Spencer is central too. I'll just say that right at the outset. So what are they? They're tiny extracellular vesicles. They're about the size of a COVID virus, as it turns out. They're involved in long-range signaling between cells, but they have this ability to protect therapeutic cargoes, and this is why the pharmaceutical industry in general, and also why I got interested in them, because I could see that they could protect our peptides. They have some other nice qualities. They're generally non-immunogenic. They don't, if you put an unprotected peptide in an animal, you're going to get an immune reaction eventually, particularly with repeated doses. But if they're cloaked in an exosome, that's not going to happen because the exosome flies under the immune radar. They also have an ability to cross biological boundaries, like the blood-brain barrier, for example. So just to extend the metaphor from gap junctions, which I described as a cellular internet, we can almost think of exosomes like the cell's FedEx surface, talking to its neighbour at some distance. All right. So, I forgot to use one of my props. So at this point, I'm going to give you an example of another one of their qualities. So milk is extremely rich in exosomes. That's good. So I just drank 1 trillion exosomes. 1 trillion. And of course, I'm not lactose intolerant. I don't think so. Maybe talk to Sherry. But they're safe. They're safe for human consumption. But more importantly, they can be ingested orally. A lot of they're unique amongst exosomes that you can isolate from various settings, cultured cells, blood, whatever. But milk exosomes have this ability to be taken up orally and then transfer from the gut into the blood circulation. Now we had this particular problem though. We had to figure out how to get them out. We knew that there was a lot of them in there, and this is the problem we faced. So the milk, they don't sit. So exosomes don't exist like little spaceships floating around in the milk. They're actually tethered to a complex of proteins, caseins, that form a kind of matrix. So part of the task that we had to set ourselves was how to separate the exosomes from the casein matrix. And to do that, we used this molecule here, EDTA, which broke up the casein. We have another way of doing it now based on the amino acid tryptophan, but at the time when we first started, this is the way we detached them so we could collect them. We also avoided using the ultracentrifuge, which is a specialised machine. The problem with the ultracentrifuge is you can only do small batches of fluid at a time. But we wanted the ability to scale from the lab bench, which is about 1 litre, up to tens of thousands of litres, because what we wanted was a way to industrially produce the exosomes. So we developed a technique that was based around filtration steps. This one here, tangential flow filtration, Sepharose column filtration, and eventually, starting from around 1 litre of milk, we get down to 100 mils of exosomes, purified exosomes that look a bit like this. Each one of these little bubbles here is one of our exosomes that we've managed to pull out from the milk. The rest of the slide is characterization per colleagues in the field, including the types of proteins they carry, their size, etc. But this is a technique that we developed ourselves to figure out what proportion of the exosomes were still intact. Obviously, if you isolate them, you don't want them to be leaky, because if you're loading drugs in them and they're leaky, they'll leak right out and they'll be no use to you. So this was based on a dye called calcine AM. It's an ester dye. And as I'll tell you in a minute, this trick with the ester dye turned out to be real important to us. So a couple of bullet points here. So these ultra-dense concentrates account for about 10 to 15% of the input volume of the milk. So if I'd filled this glass up, that much of it would be exosomal. That just gives you an idea of, the kind of density of these little structures that are present in milk. And the numbers are astronomical. There's a quadrillion exosomes in a litre of milk, a quadrillion. That's a huge number. Spencer went down to New Zealand, where originally I'm from the south, as you can tell from my accent. And he worked with a large dairy concern down there, Fonterra, in their pilot plant. They basically gave him a tanker full of milk, and he was told to go at it. And he came back from New Zealand with kilograms of freeze-dried exosomes. And when I say freeze-dried, this was another innovation that we moved into, and Spencer was central to this, but also a very talented medical student who worked with us, Alan Dogan. Alan just graduated with a letter of distinction, in part due to the work that he did in our laboratory. Just extremely hardworking, creative, again, another remarkable individual. This is our wonderful technician, Claire. She's holding a bag full of freeze-dried exosomes, and here they are here. We had a little bit of problem getting this white powder through customs back from New Zealand, but we eventually managed to get it back. Basically, we figured out a way of combining the sugar as a cryoprotectant and this amino acid tryptophan, which gave us an ability to freeze-dry these exosomes, and so we could store them at room temperature for up to a year. And I think the last time we looked, we were able to keep them at room temperature up for 18 months, I believe. Now, as I mentioned, a big problem that we also had to solve was how to get our peptide inside them. You know, the membrane is a barrier to charged molecules like small peptides. And I told you about the calcine dye with its esterified groups on it. Basically, this is how we solved it. So calcine is a fluorescent molecule, but they manufacture it so it has these ester groups linked to it. And what happens is the ester groups make the dye molecule soapy so that it moves through the membrane, and then once it gets inside the cell, the ester groups get clipped off, trapping the glowing molecule inside the cell so it can no longer get out. And so I said, well, why can't we just do that with the peptide? I mean, why can't we just make it a soapy little molecule that can get inside our exosomes, just like our dye molecule calcine? So to achieve that, we worked with Rachel Lettieri, a talented chemical engineer at UVA and a Ph.D. student at the time, Mark Bannon. And she put these ester methyl groups on our peptide. And Mark used mass spectrometry to show that we were getting inside the exosomes. And this is another method that we use that was pioneered in my lab by another talented student, Rahul Amin. Rahul was able to show that the exosomes could be loaded with a fluorescent esterified peptide molecule in a dose-dependent manner, such that 95% of the exosomes now contain the peptide. So, we're able to produce the exosomes at scale, we're able to freeze-dry them and store them, we're able to load them with our peptide. The next job was to figure out, can they help with radiation injury? So, to do this, we started collaboration with Electra Athanasiadi, who's a veterinary oncologist who works just across the road here. I don't know, is it Electra, are you here? She may be online. She probably put her hand up online. You know, this is not a great experiment, but in a sense that, you know, the mice actually have to die during it. But we subjected mice of about 15 per group to doses of lethal radiation. And what we found was that in the control groups, i.e. the irradiated control group, or mice receiving exosomes not loaded with the peptide, or treated with the peptide but not the exosomes, the mice all died by the 7th day of the study. The 14 gray dose is a very heavy dose of radiation. But remarkably, the animals that received the peptide-loaded exosomes survived at about 42%. And we're told, and we find from the literature, that there's just nothing else that really reports such a high level of protection from a heavy dose of radiation. Moreover, we found that we could, in this case, the treatment was 1 hour after the lethal radiation, but we could delay the treatment up to 24 hours after the radiation and still get significant survival, a little bit less than the 42% that we saw for the 1 hour treatment, but nonetheless statistically significant. We also looked at how it had protected the tissues and organs of interest. So, when people get exposed to lethal radiation, I don't know if anybody saw the TV series Chernobyl, where you had these brave Russian firefighters going in and fighting the radiation fire at the nuclear plant. All of them died of problems associated with the gut. And you can see why. So in the guts of mice that had passed away during the experiment, you can see that basically they've lost the structure of the gut. The absorptive villi, the surrounding muscle are basically broken down. But by contrast, the animals that had received, at the end of the study, the animals that had received the peptide-loaded exosomes, both at 1 hour and 24 hours, showed nice preservation of their gut structure. And importantly, they also showed preservation of bone marrow. You can see here these sort of dark purplish cells, or the bone marrow cells. You can see they're completely absent in the irradiated control, but the treated animals maintain them. We had a veterinary histopathologist look at our samples blind, and sure enough, the treated animals were not statistically significantly different from the non-irradiated control animals. So we had evidence of preservation of gut tissues from the experiment. Now, you've probably heard of the reproducibility crisis. It's well known that many experiments that are carried out in research settings often fail to be reproduced when they go to a pharmaceutical company or the like. And so at this stage, we were fortunate enough, supported by the Red Gates Foundation through the gift that Mike had organised, to fund a clinical trial. Of course, this wasn't going to be a clinical trial on humans. You can't test lethal radiation on humans. But the FDA has a special pathway called the FDA Animal Rule for indications that are lethal and also of strategic interest to the United States. And so we went to the FDA, we told them our story, we showed them our data, and they said, yes, you can go on this pathway. You need to do 2 animal species, you need to get data on the drug uptake and breakdown, what's called pharmacokinetics, you need to get toxicity data, et cetera, but this pathway you can use. So now this is the scary part for us. We had to basically transfer our experiment out to a contract research organisation. And of course, they don't care whether the experiment fails or not. All they care about is they get their money. And so the Red Gates Foundation, of course, funded this very expensive series of clinical trials. And we've just completed the first part of this clinical testing. We did what was called a dose ranging study, where we looked at 6 different doses of the exosomal therapeutic. Of course, the trial is blinded and randomized, and we looked at the effect of the doses relative to no treatment or unloaded exosomes. You'll also see that the study involved a slightly lower dose of radiation than the 14 grade that we used, but it would result in a predicted 70% mortality over the 30 days of the study. There were 20 mice in each group. They were balanced for males and females, and they each received an oral gavage. And so I'm happy to report that basically we were able to reproduce what we saw in the lab. So we avoided the reproducibility crisis. This is the standard dose that we use, and this is 20% of the standard dose, and this is half of the standard dose. And the beautiful thing about it was, and here you can see we've got about 65% of the untreated animals expiring over the course of the study, which is what we'd expect from the 70% lethality. It's within 5 to 10%. But 100% of the males survived. Only one of the females died in the 0.2 group. And there really wasn't too much difference between these three clustered doses very close together. So next up, we have the second phase 1 part of the study, and here we'll be testing repeated dosing and also looking at delayed timings to see if we can hone in on a regimen that's useful to cancer patients and also as a medical strategic countermeasure. And of course, those regimens may vary according to those two different needs. And then the phase 2 of the study will be using those optimized doses and regimens in pigs, which is our second species that we've looked at. So, you know, this has only happened in the last week or so. We got this data, and, you know, I was a pain in the ass in the lab, you know, marching around saying that, you know, we need to get the quality control, because we were making the exosomes and sending it to the CRO, and I didn't want us to be the one to mess up. And so it was with great relief when we started to see the clinical trial results coming through. Now, as I mentioned, milk exosomes have this unique ability to be taken up orally and biodistribute throughout the body. And this was a nice study done, not by us, but by another group a few years ago, showing that this oral route results in relatively uniform distribution in major organs and tissues, including the lungs, brain, spleen, kidney, heart, etc. Well, we wondered about this uptake, and so we basically confirmed similar uptake in the brain, the gut, the liver. But what we noticed was that in animals that were irradiated, there seemed to be an increase in the uptake, suggesting that there was some sort of injury-associated increase in the uptake of the exosomes. We measured it, and it was statistically significant. And at this point, I'm going to pivot a little bit, but it's still basically the same story. We wondered whether this was a general characteristic of injury. That does basically do milk exosomes, when they're ingested, know to go to where the trouble's at, to go to injury, to go to diseased tissue. And so to address this further, we worked with another couple of colleagues at UVA, Antonio Abate and Stefano Toldo. Stefano is expert at giving mice heart attacks. He ties up one of the coronary arteries and generates an infarct. And sure enough, in the infarcted left ventricle, we saw this very dramatic surge in the uptake of the exosomes. If you looked in the uninfarcted right ventricle, there was much less uptake. Now, injury to the heart is associated with a release of a molecule from the cells called ATP. You may have heard of ATP as the part of the energy machinery of the cell, but it also has an important signaling role. And so we treated these mice at the same time as they received the infarct with an enzyme which basically removes the ATP by breaking it down to ADP. And when we did that, we lost this profound increase in the uptake of the exosomes, as you can see here. Now, we measured it, and here is the increase in the infarcted myocardium. Notice this is a log scale, so each step here is a 10-fold change. So the distance between here and here is a big one. Here's the level of uptake in the infarcted left ventricle, and here is the uptake in the remote right ventricle. And these are uninjured controls and mice that received the surgery but didn't receive the infarct. In all cases, we got very striking significant differences between these groups. And then importantly, here's the apyrase treatment. You can see that it's just dropped away to virtually nothing. So we also looked at the surgical skin, the skin associated with the surgery where we were getting to the heart for the infarct. And we also noticed that this mechanical injury resulted in an increase in the uptake of the exosomes. Not quite as striking as we see in the heart, but nonetheless, again, suggesting that mechanical injury may be associated with increasing the uptake of the exosomes. So again, just to repeat, the exosomes seem to know to go where the trouble's at. And it seems to be driven by the extracellular ATP. We took this into a culture model. This is Claire Beard, a wonderful technician in our laboratory, works very closely with Spencer. And basically what we did was we, again, grew a lawn of cells, put a scratch wound in it, and then looked to see whether the scratch wound was associated with an increase in the uptake of the exosomes. In this case, the exosomes have been labelled with a fluorescent dye, as they were in all the other studies. I'm sorry I forgot to mention that. But importantly, when we added this enzyme, which breaks down the ATP, we got a very profound loss of the uptake increase, basically recapitulating what we were seeing in the living heart and the living skin of the mice. We measured it, and everything worked out statistically. Wounding increased uptake, a pyrase reduced the uptake in the presence of a wound. We also put ATP on unwounded cells, and again we saw increases in uptake, suggesting that ATP was in the frame as the key molecule for this response. But the experiment I want to tell you about is this one here, where we mixed the exosomes together with ATP in a test tube, and then rigorously washed off the excess ATP, so that the ATP treatment was confined to just the exosomes. The cells didn't see it. We call this ATP priming. And when we did this, we saw the uptake increase again. And so what it teaches us is that the ATP is not affecting the cells, it's somehow modifying the surface of the exosomes to make them more likely to be taken up by the injured cells. So milk exosomes literally don't home to injuries. They don't sniff injuries out and go there. Basically what's happening is that they're guided by extracellular ATP concentrations, which occur locally in injured and diseased tissues. And so that's how they know to be taken up. Well, they don't really know because they don't really have thoughts, but you get my drift. Okay, so you may be asking yourself, Rob's doing all this work in injured heart with his buddies up at UVA. What's the effect? And he's seen these great effects in the radiation model, but what about heart attacks? Isn't that a far more important injury to be focusing on in terms of human health? But we did know from a previous study, this is Jenny. Jenny did a Ph.D. with me a few years ago. She returned to China, where she is a senior pediatric cardiologist in Shenzhen now. But she very beautifully showed in isolated hearts, remember I told you at this stage we couldn't put their peptide into the living animals because it would break down too quickly, but we could isolate hearts, keep them alive and beating, and then subject them to an ischemic injury and treat them with our peptide. And she was able to show that the peptide protected the hearts against ischemic injury in this context. So again, we worked with Stefano and Antonio, but now, of course, we had the ability to protect the peptide and give it as an oral treatment. We oral gavage the mice with peptide-loaded EVs, but we also had an interesting twist on this experiment. We didn't just give them directly after the infarction, we waited an hour or waited 2 hours, in the same way that we waited 24 hours in the irradiated context, and then asked how serious was the injury to the heart that resulted from this ischemia and reperfusion. So this is the case in which the mice received exosomes that didn't have peptide in them, and this sort of white swath here is the infarct. It's the dead muscle cells associated with the ischemic injury, and you can see it's quite big. These are mice that didn't receive anything except the vehicle in which the exosomes came in, and again, the injury is big. But look here, 1 hour after infarct, we can barely see the white tissue that marks the infarct. And even if we wait out to 2 hours, the infarct is significantly smaller. And when I say significant, we've actually measured it, and we see about a 60% reduction in the size of the necrotic injury to the heart if we give the exosomes loaded with the peptide directly after we close the animals up from the surgery. And it's not too much different with an hour delay. But if we wait 2 hours, we still get a significant reduction in infarct. So why is this important? Well, most people don't know whether they're going to have a heart attack, right? And it's hard to treat them immediately. But if you have a window, a couple of hours after the actual heart attack, then you have a chance to give them a therapeutic which will save some of their muscle and prevent the injury having serious effects on the patient, including heart failure is one of the biggest sequelae that roll out from a heart attack. We also used echocardiography to look at the function of the heart, and again, we're able to establish that even in the delayed treatment, the function of the heart was recovered. So Mike mentioned the translation of work from the Institute out into companies. Spencer and I are working hard on the development of this particular company, the Tiny Cargo Company, and so wonderfully supported by members of our community. Eternally grateful to the folks that have gone in behind us. But this company will have an important role in the clinical development of the exosomal therapeutic going forward. And that will include these clinical trials. So of course we're going to, the first study is a safety study in humans. Of course there'll be no lethal radiation here. We'll simply be giving the exosomal therapeutic to healthy humans and seeing whether they tolerate it well. And we heard from the contract research organisation that the mice showed no adverse effects. They were very happy with the lack of side effects of the treatment. We haven't got the official report on it yet, but the treatment looks safe. This will be funded by the Red Gates gift, we hope. I hope we'll have enough money left for that. But what's not covered at the moment are the ongoing trials for the dual use, the trials for the dual use in cancer radiation therapy. And this will take phase 2 and phase 3 efficacy and safety clinical studies of the therapeutic and looking at its effect in mitigating radiation side effects. We haven't chosen the cancer yet. We're thinking that probably cancer of the gut, patients treated with radiation therapy for cancer of the gut would be a good target. But that's still very much in the work. And of course, we're going to be doing preclinical and work in pigs, clinical work in humans eventually for the heart attack indication. And I also just want to mention we're working hard on the cellular and molecular mechanism. And I have a wonderful postdoc in my lab, Minjong Kang. I can see Minjong up there. And she's trying to figure out the mechanism at the cellular and molecular level of how this peptide mediates its effect. And, I can assure you that she's making some very interesting progress. Not ready to talk about it yet, but nonetheless, it's great work. The Tiny Cargo Company is also very much involved in the good manufacturing practice, which is a term of art, generating a clean room for the first steps in the exosomal isolation process. They have leased a building in Foogate Road, and they're assembling the plant that will do these steps. There's also a clean room at the Institute for the final few steps of the exosomal therapeutic that was funded again by the Red Gates Foundation, and also a grant from Go Virginia, and also support from Mike as well, which I'm very grateful for. So that's the end of the story. I'm sorry I've gone over time. Thank you for being so patient and sticking with me. Again, I just want to say so much thanks to my lab, also to the NIH for my funding, as well as the Fralin family, the Red Gates Foundation for the support of our work. And also I want to say a very special thanks to some other remarkable colleagues that have worked with me. This is Laura Beth Payne. Laura Beth and I co-lead my laboratory. She's a research assistant professor at the Institute and just an amazing colleague. Linda Collins has an office next door to mine, and so she keeps me in shape and keeps me moving. But she is the administrator of the centre, and it's just I'm just privileged to have such a wonderful colleague so close by. I also want to say thanks to my former lab manager, Jane Jordan. Jane put up with me for nearly 25 years. We started working together at MUSC in Charleston, and she actually was the one that said to me, Rob, you should look at exosomes in milk. I think it's going to be a pretty rich source. Great colleague, and miss her a lot. She was tough though. She never cut me an inch of slack. Maybe that's a good thing. And then finally, I want to thank the wonderful administrative folks at our institute, embodied by Sherry Cook, who leads a team. You know, I always hear from other colleagues from other parts of Virginia Tech how amazing our administrative and grants team is, and you know, they're a credit to Sherry. Many of you probably also know that I have a conflict of interest when I'm talking about Sherry. So that's the end of the story. There's some disclosures, which I should have put up at the beginning, but you can look at them now, and I'll be happy to answer any questions.
Thank you very much, Rob. In light of the time, I think I know a bunch of people have to get to various meetings. We'll let people break, but if you have questions, please come up to the front. I'm sure Rob would be glad to. So please join me again in thanking Rob.